Hello students, we have started with the chapter number 5, that is cell, the fundamental unit of life of class 9. In the previous session we have seen the discovery of cell and how the cells functions within the body. Today we will learn the structural organization of the cell. That means how the cell is organized, how different organelles are present in them, how it functions, all things which is present inside the cell and how it is organized, that we will study. And before that we will understand about the cell membrane. Okay, so what is cell membrane? Cell membrane is the outermost layer around the cell that separates the internal part from the external environment. Suppose this is the cell, this is the fluid of the cell, these are some organelles, you can say. This is the nucleus, these are some organelles in the cell. Cell membrane separates these organelles or the cytoplasm from the external environment. That is, it is a kind of layer surrounding the internal matter. Okay. This cell membrane is made up of protein and lipids. What the substance from which the cell membrane is made up are protein and lipids. So this cell membrane, again, this cell membrane, as we can see over here, is made of bilayer. Bi means two. So these are the two layers. It is in the two layers present into the cell membrane, and the cell membrane is also termed as semi-permeable membrane. Either we can say cell membrane or also semi-permeable membrane. Now what is permeable? Permeable, something like allowed to enter. Say for an example, when you go at home, do you need to take any permission to enter in your house? No, because you are permitted to enter in the house. But if some stranger is roaming around in your house, ring the bell, would your family member will allow them to come in? No. So they are not permeable. Here the same way, cell membrane will allow only those substances which are needed by the cell. That means it will select the thing which is allowed to enter into the cell. That is why it is called the semi-permeable membrane. Correct? Now we'll see the structure of the cell membrane. As I have said, this cell membrane is a bilayer, and this layer, each layer is made up of phospholipid. That is why it is called the phospholipid bilayer. Okay, within the phospholipid bilayer, lipid bounded with a phosphate big bond. That is why it is called the phospholipid. This is the phospholipid bilayer, and in this bilayer there is the presence of some proteins. As I said, cell membrane is made up of the protein as well as the lipid. So the lipid part over here is a phospholipid, and it is made up of the two layers. That is why it is called the phospholipid bilayers. And the protein which are present in the cell membrane are of two types. One are external proteins as well as integral proteins. This protein, as you can see over here, this is the protein molecule. This also the protein molecule. These protein molecules are called the integral proteins. As you can see over here, the phospholipid bilayer has head and a tail-like structure. This is the round portion over here is called the head, whereas the internal portion, it is called the tail. So the tail is hydrophilic in nature. Philic means attracted towards. Hydro means water. So this head, this round part is attracted toward water, or we can say the water is attracted towards the head. This proteins, as this head are present at the both the side, like this is the cell membrane, and some heads are here at the both the side, and this is the internal part. The tail part is inside, and the tail part is hydrophobic in nature. That is why we call the hydrophobic tail and hydrophilic head. Okay, why this so? Because they are responsible for transferring in certain essential elements inside the cell, as well as to take some substances or waste material outside of the cell. Okay. Now, how does these substances get enter into the cell? The very first mechanism they follow is called diffusion. Diffusion is a spontaneous entry or exit, or we can say it is a spontaneous transfer of material from higher concentration to the lower concentration. And what material get diffused from the cell, or what material can transfer from outside to inside or inside to outside? That is gas. So the gaseous substances which get transferred from higher concentration to lower concentration, that is called diffusion. Now what kind of gas needs to be transferred? Those are oxygen and carbon dioxide. As we breathe in, along with all different gases, the oxygen will also get enter, and that oxygen needs to reach at the cell, because all cell needs oxygen for the respiration process, right? So to perform the respiration, they need oxygen. But again, how do they transfer the oxygen inside the cell? Okay, the question is, how this oxygen molecule get enter into the cell? The mechanism for the transfer of oxygen molecule from outside of the cell to inside of the cell is by diffusion. And in the diffusion, when we breathe in, at the outer side, this is oxygen molecule. Okay, the concentration of the oxygen molecule, oxygen will be higher than inside, because whatever the oxygen molecule, oxygen present inside this cell gets utilized, that used up in the respiration process. We can say in the cellular respiration process. So the oxygen molecule is used up. Now over here, carbon dioxide molecule will be present in the higher amount. As we breathe in, the oxygen molecule will come on the periphery of the cell. Now oxygen concentration will be higher over here, and carbon dioxide concentration will be higher inside. Now what will happen? As I said, diffusion takes place from the higher concentration to the lower concentration. So this oxygen molecule will get entered inside the cell, and carbon dioxide will go outside of the cell by the diffusion method, because higher concentration from lower concentration, and here also oxygen from higher concentration to lower concentration. Clear? Okay. Then another term which is used for the transfer of substances, that is called the osmosis. Now again, osmosis is a kind of diffusion, but unlike the gaseous material, water is involved over here. So what material get transferred over here? As we have seen, the gaseous material get transferred into the diffusion, and osmosis is a kind of diffusion in which the liquid material get transferred. What liquid material over here we are talking about is some water. So the water molecule will get transferred, or we can say the movement of water. Movement of water from the higher concentration of water to the lower concentration of water. What is the concentration now? Water concentration. Suppose we make a solution. In that we take, suppose this is the beaker or glass, you can say or a tube. This is a tube. Here in this flask or glass, we take 200 ml of pure water. Okay. In this, we have 200 ml of pure water plus 10 gram of sugar. So, in which sample the water concentration will be higher? In the first one. Over here, the water concentration will be higher because in this solution, along with the water, there will be a presence of some sugar. So, the water will be higher in this than the sample B. Okay. So, if we talk about the definition of this osmosis is transfer of water from its higher concentration to the lower concentration through the semi-permeable membrane is called osmosis. Now clear the concept of osmosis. Osmosis, that means transportation of water from higher concentration of water to the lower concentration of water through the semi-permeable membrane. Okay. So these two terms are clear by which the substances get transferred, the oxygen, the gaseous substances and water molecules get transferred from either side. Now, this endocytosis. In some organisms like amoeba, they have very flexible kind of cell membrane so that they can change the shape according to their requirement. Suppose this is a cell of amoeba. When they want to angle the food particle, they will change their shape, or we can say they will change the pseudopodia and they will catch the material within themselves and get the material digested. So over here, the cell membrane is flexible in the case of amoeba. So we can say that in some organism the cell membrane is a fixed, or we can say the rigid type, or cell membrane can be flexible. Now we talk about the function. Before we talk about the function, I will show you the structure of cell membrane. As I said, this cell membrane is made up of bilayer. First layer on the top and the lower layer. So these two layers are called phospholipid bilayers. As you can see over here, the red color part, that is this one, hydrophobic tails. These are the tails of the layers, and these are the heads, hydrophilic heads. And this thing is this one white color thing present inside this bilayer, or you can say the proteins stocked inside this bilayer are called integral proteins. This make flat surfaces both the sides. If we know the structure, we can remember the function. What are the function? Cell membrane is involved in the transport of gases, or we can say the oxygen. So they can fulfill the oxygen requirement to the cell. Carbon dioxide molecule get exist from cell through cell membrane. Third one, some unicellular organisms as well as the plant cells, they absorb the water molecule through the cell membrane. That means transport of water in the cell, in the unicellular organisms and in the plant cells is through the cell membrane. Third thing, cell membrane is also involved in the transport of nutrients. Okay, nutrients, cell basically needs what? They need oxygen, water and nutrients. So the nutrient will get transferred from cell membrane, but at this time cell have to use some energy. That means it is a energy consuming process. Now we will see what happen if we put this cell in the different concentrations. For that you have to understand the types of solutions. Now we will make three different types of solution and we'll see what will happen if we put a plant or animal cell into those three different types of solution. Okay, so here to make the first type of solution, we took 100 ml of water with 0.5 gram of salt. In the second solution, we took 100 ml of water along with the 10 gram of salt. And in the third solution, we took 100 ml of water with 1 gram of salt. Here we can use either salt or sugar to make solution. Okay, so here if in this case if we put animal cell or plant cell, it will show certain reactions. But as the animal cells or plant cells are very small, and to observe those we need microscope, so that we cannot perform in the classroom. So what we have to take over here, something that we can see with our naked eyes. Suppose over here, if we take the resins, dry resins as a cell, dry resin we consider as a cell, and put in this, it will show certain reactions. So in the first solution, what will happen, here the salt concentration is very low. Here in the first solution, if you want to take the salt or sugar, you can take, otherwise just we can keep the water solution as it is. So if we put the resin in the very first solution, what will happen, after some time, or we can say the next day, it will get swollen. So we can say that the water inside is lower than the water outside. So what will happen over here, as we have seen in the osmosis process, the water will move from its higher concentration to the lower concentration. So there will be a more concentration of substances inside, but the water concentration will be lower. So what will happen, the free molecule of water will be higher in the environment. So from this matter, the water will enter inside and this resin will get swollen. Okay, so what will happen at this time? For some period of time, the water molecule will flow inside and outside from the this resin. They can either go in and out. But if we sum up, at the end of the reaction, if we calculate, the more water molecule travel inside, then they come out. Okay, so this kind of phenomenon takes place into the hypotonic solution. The solution which has higher concentration of water is called the hypotonic solution. Okay, compared to the substance which we put in. What we have done over here, we have taken more concentration of sugar or salt. Resin which we add in this, it will get shrink off. Then it will be something like this. Here you can use either 50 mg also. Why? To make the higher concentration of substances outside. That means the concentration of the water will be less. That means the free molecule of water will be less. So the free molecule of water will be not there. So now what will happen? Few molecules can enter in this. But at the same time, whatever the two molecules of water present inside the cell, inside the resin, that will come out. And as the water will come out from the cell, that cell will get shrink. Okay. So this kind of phenomenon takes place into the hypertonic solution. The third one is called the isotonic solution. The cellular concentration is similar with the normal saline solution concentration. So the normal saline, how to make? If we add 0.85 gram of NaCl salt into the water, percentage 0.85 gram salt plus 100 ml water, that means 0.85% NaCl solution is called the normal saline solution, and normal saline solution is quite similar to the concentration of the cell. Okay, so if we make this kind of solution, the cell will remain as it is. Okay, that will remain as it is. It will not get shrink or will not get expand. So this kind of solution is called the isotonic solution. Isotonic, iso, similar, both sides similar, that is called the isotonic. So the solution tonics are of three types. Hypotonic, in the surrounding environment the free water molecule will be higher. That means in hypotonic solution the water concentration will be higher outside of the cell. In the hypertonic solution, the concentration of the water will be lesser in the solution. In the third type of solution, that is isotonic solution, the concentration of water molecule will be same outside and inside the cell. That means in the solution as well as in the cell, the water molecule will be equal. So these are the three kinds of solution to observe the phenomenon which takes place into the human, or we can say the animal or plant cells. We can use some resins, or we can use de-shelled eggs. How can we make de-shelled eggs? Suppose we treat the egg with acid, hydrochloric acid, HCl, then the outermost layer, which is made up of calcium, that will get dissolved on, and only membrane will rest. And if we put this de-shelled egg instead of this resin into these three different types of solution, and if we observe the result after 5 minutes, the same kind of thing we can get. Now we will see the second component of cell, that is cell wall. But this component, this cell wall, is not present in all the cells. Do we have a cell wall in our cells? Suppose we have a cell in all our different organs. Our heart is made up of different cells. Does this heart cell have a cell wall on top of that? No, there is no cell wall. Cell wall is present in certain organisms only. Say for an example, in a plant do have a cell wall. Then fungi have a cell wall, algae have a cell wall, some bacteria have a cell wall. So certain organisms, they have a cell wall to provide them a kind of protection. Okay. So what is cell wall? It's a rigid part. Cell wall is a rigid covering layer outside of plasma membrane. Plasma membrane, or we can say the cell membrane. It's a rigid. Rigid means, as I said, plasma membrane is a little flexible. It has, though some are fixed, but then also they are flexible. Okay, but these cell wall are not flexible at all. They are the rigid outermost covering layer on the cell membrane. They keep interior components safe and intact. Second one, that prevents the water loss. How? As we said, when we keep the cell into the hypertonic solution, what will happen? All the liquid content of the cell, that will come out and it will get shrink. But if the cell have a cell wall, it will not allow the cell to do that. They will protect the cell from water loss. Third one, that is that prevent the overexpansion. Likewise, the same way as we seen in the cell in the hypertonic solution, if we put the cell in the hypertonic solution, what will happen? The cell will get expand. If more and more water it will gain, the cell will gain, what will happen? A time will come when the cell will get ruptured. But due to the cell wall, it will not allow the cell to get ruptured. Okay, when the water concentration is less, it will not allow the cell to get shrink. Okay, and it will, in the higher concentration of water, it will not allow the cell to get expansion, so that they cannot get burst off. The third one, they provide the barrier between internal and external environment. External environment is quite higher. Suppose the plants we can see in the outer side is in too much of this heat. Can we withstand that heat? No, because they have a cell wall. The cells are protected and that covering layer. That's why they can feel this environmental stress. But we cannot feel the environmental. We cannot withstand with those environmental stress as the organism who have the cell wall. Okay. It gives the protection against the environmental shock. Again the same thing. As I said, cell wall are of different type of in different organisms. In plant, cell wall is made up of cellulose, pectin, lignin and cutin. In algae, the cell wall is made up of cellulose and glycoprotein. Here the glyco word, as you see here, glyco something related with the carbohydrate or sugar. Glycoprotein, when the sugar molecule will bind with the protein, this is called the glycoprotein. Bacteria have peptidoglycan. Fungi have chitin. This peptidoglycan of bacteria, peptidoglycan is a subtype of glycoprotein. Peptido, again the gly over here as well. Sugar, something related with the sugar, that is why over here something related with the sugar. So here sugar, that is carbohydrate related with the peptido, that means peptido that related with the amino acids. The peptido amino acid binded with the sugar moiety, that makes peptidoglycan. So in the bacteria, the cell wall is made up of peptidoglycan, whereas in a fungi it is made up of the chitin. This cellulose, pectin, lignin, cutin, chitin, they are all polysaccharides. So depends on the organisms, the cell wall is of different type. Now the plasmolysis. What is plasmolysis? Normally we see lysis means breaking down, but over here plasmolysis is something else. The phenomenon of contraction of protoplast away from cell wall due to water loss. Due to the contraction, the cell material will come closer. But in case of the cell which do not have the cell wall, the whole cell will get shrinkage. But in case of the cells which have cell wall, only the inner content will get shrinkage. But the outer side, the cell will look the same. If we see inside the cell, the stuff which is present inside, that is called the cytoplasm, that will get shrinkage due to water loss. But the outer side, the cell wall will be as it is. So the phenomenon of contraction of protoplast away from cell wall due to the water loss. How it happens? This is the cell membrane and this is the cell wall. If water will get lost from the cell will look as it is, but the inner content will get shrink off. That is called the plasmolysis. This will happen when the water will be lesser in external environment. That is why the water will go out. And if we keep these cells into higher water concentration, then what will happen? This cell will intake the water and the content will get expand. If the water concentration is higher, it will start getting expand. But in case of the without cell wall, what will happen? The cell will get brushed off at one particular time. But over here, this will put again some pressure back on this cell membrane. Okay. So this will, as it is expand, it will make the turgor pressure, whereas the cell wall will again put a pressure back on the cell membrane. So by putting the back pressure, they will save the cell from expansion and from getting brushed off. So in the next session, we will start with the different organelles. Okay, thank you.